Brain Shakers, welcome to the Brain Shakers Academy. Brave Alistair is here. Now, today's session, we're going to be looking at the process of mitosis. Now, we have already started a series on a cell division where we have looked at the cell cycle. And so, if you haven't seen that video, please do check for that video of the cell cycle on my YouTube channel, which is the Brain Shakers Academy. Now, today we continue with a series of a cell division, and we'll be looking at mitosis, which is just one of the processes of a cell division. Now, most of the cells do undergo the process of mitosis. Now, what is mitosis? So the process of mitosis is a process in which one parent cell divides to form two identical cells that we refer to as daughter cells. Now, cells undergo cell division to produce new cells or to replace old ones, with the majority of these cell divisions involving the process of mitosis. In humans, you will note that there are 23,000 genes, and these 23,000 genes are on 46 chromosomes. Now, the 46 chromosomes do not appear in isolation, but rather appear as 23 homologous pairs, which we refer to as a diploid number. Now, of the 23 homologous pairs, you identify that 22 pairs of those chromosomes will be referred to as autosomes, and one of the 23 will be a pair of sex chromosomes, with the XX being genetically female and the XY being genetically male. Now, the goal of mitosis is to ensure that the cell has divided into two identical daughter cells with a full set of chromosomes, and it's able to do this by going through a number of phases that are well organized and carefully ordered. Now, what are some of the phases of this process of mitosis? So mitosis basically has four distinct phases, which we refer to as the prophase, the metaphase, the anaphase, and the telophase. Now, if you read different literatures, you will also understand that some of the literatures that is published will be categorizing them as five phases of mitosis. Why? Because the prophase is often broken down into early prophase and the late prophase, which is also known as a pro-metaphase. Okay, now these phases are very cardinal in ensuring that the parent cell goes on to divide and produces the two identical daughter cells. Now let's look into more detail, step by step, and see what happens at each and every phase of the process of mitosis, starting with the prophase. So the prophase is the first phase in the whole process of mitosis. Now, during the prophase, you will notice that there is nuclear envelope disappearance, meaning that the nuclear envelope begins to disintegrate. And what this means is that the nucleus is getting ready to undergo the process of a cell division, and the chromosomes within this particular nucleus also will condense and coil themselves up, getting ready to be divided or getting ready for the process of cell division. Within the cell, you will notice that there is also what is known as a centrosome. Now, the centrosome is contained of two centrioles, and those two centrioles are very cardinal in the cell division. Why? Because the centrioles during the prophase, they begin to move to each pore. So you find that one centriole moves to one pore of the cell and another centriole moves to the other pore of the cell, and these make or allow the cell to have the mitotic apparatus, which is just part of the cytoskeleton, as it contains within it the microtubules that are responsible for making sure that the cell undergoes cell division. Now, when you move towards the later stage of prophase, you will understand, which is also obviously referred to as the pro-metaphase in other literatures, you will understand that the mitotic apparatus that has now formed has the mitotic spindles, and those mitotic spindles begin to capture chromosomes and align them at a metaphase plate, which is just an imaginary plane that is created during the process of prophase as you graduate into the next phase, which is the metaphase. That is why it is called pro-metaphase, because during that particular time the chromosomes now are captured by the mitotic apparatus, and then they are aligned at a metaphase plate, and that takes us to the next stage, which is the stage of metaphase. Now let's look at what happens in metaphase. The following phase after prophase is the metaphase, which is basically position changing. Now, what happens in the metaphase is the mitotic apparatus would have already formed. The mitotic spindle fibers as well are present in the cell. And once these ones have been formed, what is going to happen is they begin to capture the chromosomes and align them on the metaphase plate. So once the chromosomes have been aligned on the metaphase plate, they are attached to the spindle fibers and the centrioles through a point that is known as the kinetochore. Now, a kinetochore is just a disc-shaped protein around the centromere. Now, the centromere is the point of articulation of the two sister chromatids where they meet at the central part here. This is where you find the centromere. On the end of that sister chromatid, you find the telomere. Now, what we are interested in now is the centromere, which is the point of articulation of the two sister chromatids. So where they meet and come together at the central part is known as the centromere. Now, around the centromere, you will find a disc-shaped disc-shaped protein that is known as the kinetochore. So you find these mitotic spindle fibers attached to the kinetochore and getting ready for them to then be pulled apart during the process of anaphase. Now, before we get to the process or phase of anaphase, there is a checkpoint that happens during the mitotic phase. Now, this checkpoint is referred to as the mitotic spindle checkpoint, and we have a dedicated video where we have looked at the cell cycle and what happens in the different cycles of interphase and the mitotic phase. And do look for that particular video on my YouTube channel, which is the Brain Shakers Academy. And if you haven't subscribed, please do remember to subscribe as well. Now, there's going to be a checkpoint here that checks and makes sure that all the chromosomes have been aligned onto the metaphase plate, so that when the cell begins to divide and there is that pulling effect during the anaphase, the sister chromatids are going to separate and will be in equal number for the two daughter cells that are going to form. Now, if there is any chromosome that has not yet been aligned to the metaphase, what is going to happen is that the cell will not proceed into anaphase until that problem has been fixed and until that chromosome has been captured by the mitotic spindle fibers and aligned on the metaphase plate. Remember, we said that the entire process of mitosis or cell division is a well-ordered and organized process, making sure that the two daughter cells that are going to be produced from one parent cell are identical and they possess within them the equal number of chromosomes. So that checkpoint makes sure that all the chromosomes now have been aligned on the metaphase plate and getting ready for the next phase, which is the anaphase. Now, let's look at what happens once we have all the chromosomes aligned on the metaphase and then we have moved into the next phase, which is the anaphase. Now, during the anaphase, you find that a furrow begins to form in the cell membrane. Now, why is a furrow forming in the cell membrane? This furrow begins to form because the centromeres or the centrioles that were on the end or in the pores of the cells would have then pulled the sister chromatids. So this is a sister chromatid, this is a sister chromatid. So these are chromatids. Now, when they were together, they were attached together at a central mere, and then they have been pulled together because of the mitotic apparatus effect, where there is contraction of those microtubules. And remember, we said that the mitotic apparatus is part of the cytoskeleton and has a contraction effect, and that contraction effect pulls the sister chromatids apart from one another by binding to their kinetochores. So one of each pair of the sister chromatids then migrate to the pores of the cell, one to the left and the other one to the other side as well. And so the microtubules that are part of the uh, mitotic spindle are the ones that are responsible for the movement of the sister chromatids to the pores. Now, you will find that there are mitotic fibers or mitotic spindle fibers that are not attached to any chromosomes. So what these mitotic spindle fibers that are not attached to any spindle fibers do is that they will begin the process of elongation, and as they elongate, it means that they pull the centrioles far apart, creating more room and space in between the sister chromatids, allowing a furrow to nicely form, and so that when the cell then divides in between, the two daughter cells that are going to be formed will contain the same number of chromosomes within them, so that there is no chromosome that will be caught along the line of separation. And then that takes us to the telophase, where now there's going to be disintegration or separation of that cell that has elongated into two cells. Now, what happens in the telophase is the mitotic apparatus that had formed during the prophase and got the cell ready to separate into two daughter cells has completed its job and has now separated the cell into two. Now the remaining part is for the cytoplasm to obviously break down and divide into two daughter cells. So the mitotic apparatus will then begin to disappear and will be broken down into its building blocks. And you also notice that the chromosomes that had condensed and coiled themselves up in readiness for cell division will begin to uncoil and to decondense themselves in readiness for either entering into a permanent state or getting into another cell cycle. Then during the prophase, the nuclear membrane had disappeared, so that the nucleus can then go ahead and continue with the process of a cell division. When you get to the stage of telophase, the nuclear membrane now is going to reform. So it comes in and surrounds the two cells that have separated, or the two chromosomes that sets of chromosomes that have been formed in each cell. So that is what happens within the telophase, and this then prepares the cell to be completely divided through the process of cytokinesis. Now, when we get to the process of cytokinesis, it will just be the division of the cytoplasm. So that furrow that had started to form will now complete the process of separation, and there is a division of that cytoplasm into the two new cells. And you notice that the process of cytokinesis oftentimes overlaps with the final stages of mitosis, and it may even start either in the anaphase or in the telophase, depending on which particular cell. Now, in animal cells, cytokinesis is just the contractile pinching of the cell into two daughter cells through that cleavage furrow. And the organelles that are formed in this particular new cells that have been produced as a result of mitosis, they are not fully developed. And this is why when the cells have been produced, they have the capacity to enter into the cell cycle again, which is the whole process of interphase, and then get to the mitotic phase. Now we have a dedicated video as well on the cell cycle, and if you haven't looked at that one, please do make sure to look at that one, and I'll put the link as well in the description below so you have quick access to it. So that is the process of cytokinesis. So once the two daughter cells have been formed, the process of mitosis is now complete, and the two new identical cells or daughter cells that have been formed contain within them the equal number of chromosomes and the relevant organelles that will need to then be matured, and will need to then get into the right amounts and size for the cell to then complete another process of cell division. So the cells can either get into an interphase or get into another cell cycle, or they may get into what is known as the gap zero or as resting phase if they are not actively or rapidly dividing cells, and then the cell repeats itself. This is about the process of mitosis. Now all you need to remember is that in mitosis you have four distinct phases, that is the prophase, the metaphase, the anaphase, and the telophase, and these processes are well coordinated and ordered, and at every phase they are checked to make sure that the cell is undergoing the correct procedures to make sure that the resulting daughter cells are identical and possess the correct amount and number of chromosomes with good integrity. Now if you did like this particular video, do not forget to subscribe to our YouTube channel, which is the Brain Shakers Academy. Please give it a thumbs up, like and share as much as possible. You may just be helping somebody else to understand the process of mitosis in simplicity. As always, it is a pleasure to have you here, and thank you for watching. I'll see you in the next one.